Hi everyone, I am Mr. Sia, biology teacher from SMK Luar Bandar Nomor 1 Sibu. Today, we are going to spend 1 hour 30 minutes together for the discussion of multiple choice question in biology paper 1. I will carry out the discussion for the first 45 minutes and then Madam Asmaizan, biology teacher from SMK Agama Sibu, will continue to discuss another 20 multiple choice questions with you all. So for your information, I will keep quiet for a while after I show you the question. I want you to read and understand the question before the discussion. So please get ready your writing materials and also calculator. Question 1. Okay, so for this question, the question asking cannot substance that cannot be disposed into the sink. So there are 4 choices here. So you categorize them into the substance that can and cannot, okay? Can or cannot be disposed into the sink. We do some revision for the liquids that can be disposed into the sink. The first one is the acidic and alkaline waste of the pH 5 to 9. So weak acid, neutral, or weak alkali. Okay? So the second one is the liquids which are low concentration. They are non-hazardous. They are biodegradable. For this group of the substance, they cannot be disposed into the sink. So the first one is the volatile substances, reactive substances, substances with the pH less than 5, means strong acid, and also the substance with the pH more than 9, strong alkali, chemicals, radioactive substances, solid waste, organic waste, heavy metals, organic solvents, and toxic substances. Now we look at the answer one by one. Distilled water, yes. Salt solution, can. Hydrogen peroxide, cannot. Hydrogen peroxide cannot because hydrogen peroxide is reactive substance, is reactive oxidizing agent or bleaching agent. Rubber cannot because rubber is solid waste. It will clog the sink. So the question asks for cannot. The answer is 3 and 4. Question 2. So for this question, okay, asking the system, you have to understand this diagram. You have to know the organs that involved in this system. So the organ here, liver, stomach, pancreas, small and large intestines. Okay, so is it excretory system? No. Excretory system, kidney. Respiratory system, no, lung. Integumentary system, the main organ is the skin. So the answer is digestive system. Digestive system D. Question 3. So this bar chart shows 3 types of organelles that present in the cell P. The highest bar is the Golgi apparatus, followed by rough endoplasmic reticulum. Then the shortest is the mitochondrion. So do you still remember what is rough endoplasmic reticulum? is the endoplasmic reticulum with the ribosomes attached to the surface. So the function of the rough endoplasmic reticulum to transport protein synthesized by ribosome. Then Golgi apparatus to process, modify, pack, and transport protein, carbohydrate, and glycoprotein. So now we look at the choices given. Okay, sperm cell and muscle cell should be more mitochondrion. Okay, then the organelle that abundant in the palisade mesophyll cell should be chloroplast. We only have pancreatic cell. So the answer is pancreatic cell. If I change this question to why Golgi apparatus and endoplasmic reticulum, they are abundant in pancreatic cell. Do you know the answer? The answer is to increase the synthesis and the secretion of the digestive enzyme. Question 4. The information you can get from this question: mustard green stem, it is plant cell. And then it's immersed in 30% of the sucrose solution. So this 30% of sucrose solution is hypertonic solution, isotonic solution, or hypotonic solution. Then do you still remember what is the isotonic concentration for the cell sap of the plant cell? The answer is 5%. 5% of the sucrose solution is the isotonic solution for the plant cell. So 30%, which is higher concentration than the 5%, then it is hypertonic solution to the plant cell. So now we look at the hypertonic solution. What is hypertonic solution? Hypertonic solution is a solution with high solute concentration and also low water potential. Then now you have to apply the concept of the osmosis, where the water diffuse from high water potential region to the region with low water potential through osmosis. Okay. Then you look at the answer A and C. We categorize the answer A and C, they are the same. When you look at the epidermis and the cell sap of the plant cell or the cortex, the cortex is longer, then more water diffuse into. So more water diffuse from the solution into the cell sap of the plant cell. Then you look at C. The bending is greater. The bending is greater, then more water diffuse in. Water also diffuse from the solution into the cell sap of the plant cell through osmosis. So both of these A and C, they are immersed in hypotonic solution. And then B, the epidermis looks like longer than the cell sap of the plant cell. So water diffuse out. Water diffuse out from the cell sap of the plant cell into the hypertonic solution. Okay, so the water potential in the cell sap of the plant cell is higher than the water potential in the hypertonic solution. So water diffuse from the cell sap of the plant cell into the hypertonic solution through osmosis. Okay, so this one is immersed in hyper. And then D, okay, the length of the epidermis and the cortex, they are the same. So water diffuse in and out at the same rate. So this is isotonic solution. Okay, so D, the stem is immersed in isotonic solution. So the answer is B. Okay. Question 5. So from the question 5, we know that it is a plant cell. Okay, so do you know what is this cell? Okay, shown in the diagram. It is root hair cell. And then now we look at the direction of the movement of the mineral ions from low concentration to high concentration. Okay, from outside into the root hair cell. So against its concentration gradient, so it is active transport or passive transport? It is active transport, okay, with the use of the energy in the form of ATP. So active transport. So from the answer here, which of the following describe the movement of the mineral salt ions? Then you look at the answer A, B, C, D, absorption, active transport, okay, according to the concentration gradient or against the concentration gradient. Okay, for according to the concentration gradient must be wrong. Okay, this one's for passive transport. Active transport, yes, active transport against the concentration gradient. Okay, so this is D. Let us do the revision for the examples of the active transport. There are 4 examples for the active transport. The first one is the absorption of the glucose and amino acids in the villus. Second is the reabsorption of the glucose and amino acid, but at different place through the proximal convoluted tubule in the kidney. The third one is the transport of the sucrose from a leaf to a phloem tissue. The last one, absorption of the mineral ions by a plant root hair cell. So this active transport happens, okay, against the concentration gradient by using energy. Question 6. From the answer given for the classes for chemical substance L, M, and N, there are proteins, lipids, and carbohydrates. So do you still remember the monomers for the proteins, lipids, and carbohydrates. Now we look at the diagram. The first one, L. Do you know this is glycerol? And then these three fatty acids. So this is the first fatty acid, second fatty acid, and third fatty acid. So L should be lipid. For M, you look at the repeating unit. They are glucose. So carbohydrate is made of glucose. And then the last one, there are different shapes for the monomer. So this is amino acid. We have 20 types of amino acid. So this is protein. So the answer is D. For carbohydrates, we have monosaccharide, disaccharides, and polysaccharides. For polysaccharides, I want to show you the structure of the examples of the polysaccharides. We have glycogen, cellulose, and starch. You look at the repeating unit, the monomer, they all are made of glucose. For glycogen, it is branched polymer chain of glucose, branched. And then for cellulose, they are unbranched polymer. The polymer chain are made of glucose and then they are arranged in linear pattern. They are parallel to each other. And then cellulose can be found in the cell wall. For the last one, starch, it is glucose chain with helical shape. Question 7. So this question, recall your memory about the word equation for the hydrolysis reaction of protein. So the process here is hydrolysis. Hydrolysis is the combination of two words. First one is hydro, second is lysis. Hydro means water and lysis is breakdown. You use water to break down complex molecule into more simple molecule. So from the options given, A, B, C, and D, only A and D, you use water, okay, by using water. So for A, use water to break down polypeptide into amino acid. And then for D, water is used to break down dipeptide into polypeptide. Okay, now you compare dipeptide with polypeptide. Which one is more complex? Polypeptide, more complex. So this one wrong. Okay, so polypeptide compare with amino acid. So you use water to break down the polypeptide, which is more complex, into amino acid, which is the basic unit for the protein. So the answer is A. For the hydrolysis of protein. Now I show you two-word equations about the hydrolysis and condensation process. I use an example of protein. So for hydrolysis, we use water to hydrolyze dipeptide to form amino acid. So amino acid, they are the monomer for the protein. They are simpler than the dipeptide. Okay. While for the condensation process, we use simpler molecule, amino acid and amino acid, they combine to form dipeptide, which is more complex compared to amino acid, by removing small molecule like water, through the process called condensation. So you see here, the reactant from the hydrolysis will become the product of the condensation process. And then the products of the hydrolysis will become the reactant of the condensation process. So this is the concept for the hydrolysis and condensation process. Question 8. So this picture shows the lock and key hypothesis. So what is X, Y, and Z? Now we look at X first. The shape of X before the enzyme action is like this. So after the enzymatic action, it is still remain the same. So enzyme remain unchanged throughout the enzymatic action. X should be enzyme. Then we compare the size of Y and Z. Y is bigger than Z. Y should be the reactant. Z should be the product broken down into smaller pieces, product from the reactant. So Z, they are simpler than Y. So Y should be the substrate, Z is product. So the answer is B. So now my question, do you know what is this? It is enzyme substrate complex. Now we look at a cell cycle before we move to the next question. So a complete cell cycle is made up of interphase and M phase. So what are the phases inside the interphase? The first one is the G1 phase, followed by S phase, then G2 phase. For M phase, it is made up of the mitosis and cytokinesis. So the correct sequence of a cell cycle, we start from growth phase 1, followed by S phase, then G2 phase, mitosis, and cytokinesis. So what happened inside G1 phase? Cells grow. For S phase, DNA synthesis occurs. And then for the G2 phase, cells continue to grow. For M phase, it is karyokinesis. It's the division of the nucleus. It's made up of prophase, metaphase, anaphase, and telophase. For the cytokinesis, it's the division of the cytoplasm. Question 9. So the information you can get from the question, it is a cell division, and then it occurs in animal cell. So from this diagram, 2 daughter nuclei, okay, completely divided already to form 2 daughter cells. So which of the following is not true? The chromosomes are uncoiled and become long and thin chromatin threads. So this is the chromosome. Okay, and then you see the chromosome change to chromatin. It's the chromosome in the thread-like structure. Okay, this is correct. The centromere of each chromosome divide into 2. So centromere divide into 2 and move to opposite pole. So then this one should be for the anaphase. Okay, anaphase. See, the spindle fiber begins to disappear. No more spindle fiber. Start to disappear because the chromosome reach the opposite pole. No more use for the spindle fiber, yes. And then this is cytokinesis. Cytokinesis occur, yes. This is the division of the cytoplasm. It is cytokinesis. So the answer is B. Question 10. The diagram below shows the setup of experiment that use yeast. Which of the following equation represents the result of the experiment above? So we have 2 sets of the experiment. In the boiling tube A, we have 5% of boiled glucose with yeast suspension. And then for boiling tube B, only 5% boiled glucose solution without yeast, no yeast. So this is alcohol fermentation, a type of anaerobic respiration. Now you look at this one, paraffin oil. Why you use paraffin oil? To make sure the fermentation of yeast occur without oxygen. The oxygen from the atmosphere cannot dissolve into it. And then why we use boiled glucose? We want to eliminate the dissolved oxygen inside it. So this is anaerobic, so cannot with oxygen. And then for C, with energy, also wrong. And then only left D. Now, the products are carbon dioxide, ethanol, and energy. How are you going to test the presence of the carbon dioxide? The lime water will turn cloudy. Then how about ethanol? From the smell of the ethanol. And then the energy, that means this reading for the thermometer will increase. Question 11. The diagram shows the gaseous exchange happen at the alveolus and blood capillaries. And they ask you to compare the blood at R and the blood at T. The blood at R is the blood before the gaseous exchange, and then the blood at T is the blood after the gaseous exchange. You should know the partial pressure of oxygen in the alveolus is higher compared to the partial pressure of oxygen in the blood capillaries. So oxygen will diffuse from the alveolus into the blood capillary through the passive transport. While for the partial pressure of the carbon dioxide, then it is lower in the alveolus. Lower partial pressure of carbon dioxide compared to the partial pressure of carbon dioxide in the blood capillaries. So carbon dioxide will diffuse out from the blood capillary into the alveolus through simple diffusion, so that carbon dioxide can be expelled out from the lungs. So now we look at the options for the R. High partial pressure of oxygen at R, RON, should be lower. And then more oxyhemoglobin, also RON, should be less oxyhemoglobin. Then high partial pressure of carbon dioxide at R, correct. And then at T is low partial pressure of carbon dioxide, correct. And then the color of the blood at R is bright red. If it is bright red blood, then should be oxygenated blood. This is oxygenated blood. And then the color for the blood is bluish red. Then this is deoxygenated blood. So, is it oxygenated blood at R? Should be wrong. It's deoxygenated blood. Okay, then these two should be terbalik already. This is the setup of the experiment to find the energy value of a food sample. So I want to show you the formula for the energy value of food is equals to the water mass times 4.2, which is the specific heat capacity, and times increase in the water temperature, divided by the food sample mass, divided by 1000. If you divide by 1000, then you will change it to kilojoule. Question 12. Can you apply the formula you learned just now? The formula to find the energy value of a food. You may start now for the calculation. Can you get the answer? So the energy value is equals to the mass of water times 4.2, the specific heat capacity, times increase in temperature, 60 minus 28, and then divided by the mass of food. So can you get the answer? 448. The unit is joule per gram. Answer C. Now we look at the digestion of protein. Digestion of protein start from stomach. So pepsin will hydrolyze the protein into polypeptide at the duodenum, and then the trypsin acts as the enzyme, hydrolyze the polypeptide into the peptides. The erepsins in the ileum hydrolyze peptide into amino acid. So amino acid is the monomer for the protein. So the digestion of the protein ends with amino acid. Question 13. For this question, you have to look at the diagram. So this is what organ? This is stomach. And then if the stomach does not function properly, then digestion of which of the flowing substances will be affected. Okay, we learned just now the digestion of the protein start in the stomach. So mostly affected it will affect the protein. And then we have B and D. So will it affect the digestion of the carbohydrate? No. Because the presence of the hydrochloric acid, okay, hydrochloric acid in the stomach will stop the enzymatic action of the salivary amylase. So there is no digestion of the carbohydrate in the stomach, only protein. The answer is B. Okay, now we look at this one. What happened to the blood after centrifugation? So if the blood is being centrifuged, then it will separate according to the density. So now you can see 55% of the total blood is made of plasma. And then erythrocyte is 45% at the lowest part. And then in the middle is the leukocyte and the platelet. Question 14. The diagram below shows main components of blood. What is the component G? After centrifugation, the blood is separated into two layers. First layer is the plasma protein, 55% with lower density. Then what is the component G in this layer with higher density? Is it hormone, erythrocyte, enzyme, or nutrients? It is erythrocyte. The answer is B. We continue with the health issues related to blood clotting. Other than hemophilia, can you name me the other two health issues related to blood clotting? They are thrombosis and embolism. Now we look at hemophilia. What is hemophilia? Hemophilia is an example of an illness prevent the blood from clotting. The blood cannot clot. And then it is hereditary illness passed from one generation to the next generation due to the lacking of certain clotting factor, no clotting factor. And then it will cause excessive bleeding and death. For thrombosis, it is the formation of the blood clot. And then this blood clot is named as thrombus. Thrombosis happens as a result of the damage in the blood vessels or sluggish blood flow that causes the clotting factors to accumulate. For embolism, when a blood clot is transported by blood flow, the blood clot is called embolus. If the embolus gets stuck in a tiny blood vessel, the blood flow will stop. Now we move to cardiovascular diseases. We focus on two cardiovascular diseases, which are atherosclerosis and arteriosclerosis. For the atherosclerosis, the first one, athero, is the formation and the deposition of plaque. Formation and deposition of plaque on the artery walls. The plaque is formed from cholesterol, lipid, dead muscle tissues, and coagulated platelets. The plaque will clot and narrow the lumen in blood vessels. Atherosclerosis is the early stage of arteriosclerosis. So normally atherosclerosis first, then arteriosclerosis. So arteriosclerosis occurs when the calcium is deposited on the plaque and causes the artery to become hard. It is hard and less elastic. Question 15. You need to choose the correct disease based on the description given. So the blood does not clot. This is important. Even though the injury is very minor, and then It is because lack of clotting factors in the blood. And then the third one is that it is hereditary disease. So it refers to hemophilia. Answer is C. Do you still remember what is thrombosis? What is thrombosis? Formation of the blood clot, which is thrombus. Then what is embolism? Embolus start in tiny blood vessel and then the blood flow stop. The last one, atherosclerosis, is the formation and the deposition of plaque on the artery wall. For the types of the immunity, there are two types of the immunity, active immunity and passive immunity. For active immunity, we have two types of the active immunities. So there are natural active immunity and artificial active immunity. For passive immunity, they are the same. So natural passive immunity and also artificial passive immunity. Now we want to focus on artificial active immunity and artificial passive immunity. So vaccine injection is given for artificial active immunity. For the artificial passive immunity is obtained through the anti-serum injection. Now we look at what is vaccine. Vaccine is the suspension of the weakened or dead pathogens, while anti-serum is the serums contain the specific antibodies. Antibodies are produced by the lymphocyte for the artificial active immunity, but antibodies are obtained straight away from the anti-serum for the active passive immunity. We continue with the difference between artificial active immunity and artificial passive immunity. For artificial active immunity, after the first injection, the antibody level is still below the level of immunity. So second injection is needed. We call this as booster dose, so that the antibody level will increase and then above the level of immunity. Then we call this concentration of antibody is sufficient, is enough to fight with the disease. While for the artificial passive immunity, after the injection, the antibody level will increase above the level of immunity, but the level of the antibody will drop from time to time. Second injection is needed if the patient is still infected by the disease. Question 16. The diagram below shows the concentration of the antibodies in two individuals, P and Q, after they are given two doses of injections of different substances. So what are the substances injected into individual P and Q? We have to look at the type of the graph so that we know it is which type of immunity. So now we look at the graph for P. After injection, the concentration of antibody reach the immunity level, but it drops from time to time. So this is artificial passive immunity. And then for artificial passive immunity, it can be obtained through the injection of anti-serum. What is anti-serum? The serum that contains specific antibody. Then now we move to the graph Q. After the first injection, the concentration of antibody still below the immunity level. Second dose, which is booster dose, is needed so that the concentration of antibody will reach the immunity level is enough for fighting with the disease. So this is artificial active immunity. This can be done by injection of vaccine. So the answer is B. Question 17. The diagram below shows three types of nerve cells, P, Q, and R. Which of the following is the correct sequence of the transmission of nerve impulse? Can you identify the neuron P, Q, and R? The easiest one is P. It is relay neuron. Then you look at the cell body at the end. So this is motor neuron. If the cell body in the middle, then this is sensory neuron. So the sequence for the transmission of the nerve impulse from sensory neuron to relay neuron, then motor neuron. So the answer is R, P, Q, A. Question 18. The table below shows the information of the urine composition of 4 individuals. As you can see from the table, the individual A, B, C, and D with different concentration of urea and water content. Which individual is most likely to have been eating a lot of protein on a hot dry day? You need to break this question into two parts. The first one is a lot of protein, and then the second one is during hot dry day. So if you eat a lot of protein, hydrolysis of protein occurs and more amino acid will be formed. But excess of amino acid cannot be stored in the body. So amino acid will be broken down into urea. And then urea will be excreted in the urine. So concentration of urea should be higher. Then how about hot dry day? During hot dry day, your body needs more water. More water will be stored. So more water will be reabsorbed. So if more water will be reabsorbed, the water content will be less, lower. Okay, answer is D. Question 19. The diagram on the right shows a disorder of musculoskeletal system. What is the symptom of the disease? So we focus on this part, the joint. So this joint is swollen. Then the cartilage become thinner. So this is the symptoms for the disease for arthritis. Now we look at the answer A. The joints become swollen. Yes, and then the cartilage become thinner. So answer is A. Degeneration of muscle, no. The backbone is bent to the side, scoliosis. The bone become porous and lighter, osteoporosis. So the answer is A, arthritis. Okay, for degeneration of muscle, muscular dystrophy, I just show you the diagram. And then for the scoliosis, the backbone bend to the side. As you can see, this is the healthy bone. This is the bone with osteoporosis, becomes porous and lighter. Question 20. The diagram on the right shows the female reproductive system. So what are the processes that occur in P, Q, and R? So from the diagram, what is P? P is the fallopian tube, where fertilization of the sperm and ovum occurs here. So P should be fertilization. How about Q? What is Q? Q is the uterus, where the embryo implants here. Embryo implants here in the endometrium. So Q should be implantation. Then how about R? R is the ovary. It's the female reproductive organs that produce ovum. So process happen at R is ovulation. So the answer is D. I have finished the discussion with you all. Now you can scan this QR code and try the questions in the Google form. I hope my explanation can help you to have better understanding of some of the concepts in the biology. You may pause this video while you are trying the question. So Madam Asmaizan will continue the discussion with you all. Thank you.
Hi, I will continue question number 21 to 40. So let's start. Question number 21. Diagram below shows a plant cell. What are P, Q, and R? Okay, first step, choose the organelle that you very sure the answer. P, Q, R. Q, look at the diagram. Chloroplast. So the answer either A or B. Then look at P and R. The shape of P, Golgi apparatus. So R is smooth endoplasmic reticulum. So you can get the answer. The correct answer is B. Question number 22. Figure below shows a cross-section of a leaf A, B, C, D. Which of the cell label A, B, C, and D does not contain chloroplast? Figure show the cross-section of leaf. So A, epidermis, B, palisade mesophyll, C, spongy mesophyll, and D, gut cell. Okay, so the question, which of the cell label A, B, C, and D does not contain chloroplast? Okay, how to choose the correct answer? From our knowledge, palisade mesophyll are packed with chloroplast to trap sunlight for photosynthesis. While gut cell at the lower layer of leaf, green in color, also contain chloroplast. Spongy mesophyll contain very few chloroplast. So epidermis is a single layer of cell that has no chloroplast. So the answer is A, epidermis. Okay, we continue question number 23. Diagram below shows an experiment of photosynthesis. 60-watt bulb, boiling tube, water bath 37 degrees Celsius, gas bubble, Elodia, pond water with sodium hydrogen carbonate 0.5%. Which of the following will decrease the rate at which gas bubbles are released in the experiment? Okay, decrease the rate. Gas bubble released. Decrease. Okay, the word decrease. So we must choose the factor from answer A, B, C, D, which decrease the rate of photosynthesis. Okay, the first one, A. Using 40-watt bulb, lower light intensity. Okay, so the rate of photosynthesis, low. B. Using a water bath of 35 degrees Celsius. 2 degrees Celsius of water bath decrease. Okay, so the rate of photosynthesis also decrease. Okay, for answer C, replacing Elodia with Hydrilla. Both are aquatic plant, so does not affect the rate of photosynthesis. D, using sodium hydrogen carbonate solution 1.0%. The higher the concentration of hydrogen carbonate, the higher the rate of photosynthesis. photosynthesis. So, C and D, wrong answer. Okay, either A or B. Which one is more accurate? Okay, so A more accurate. So the answer is A. Number 24. Diagram below shows the structure of chloroplast. Okay, seen under an electron microscope. X and Y. Okay, look at X and Y. So you must identify X and Y. What is X and Y? Okay, X, stroma. Y is granum. Okay, so stroma, dark reaction. Okay, the dark reaction, while Y is granum, light reaction. Okay, so in the dark reaction, synthesis of glucose, while light reaction, photolysis reaction, or we call it as photolysis of water. So the correct answer is B. Diagram below shows organism P, Q, and R. What are the type of nutrition for organism P, Q, and R? Okay, P, at that trunk, mushroom, so saprophytic. Q, epiphytes. Okay, R, rafflesia. Okay, we know rafflesia is parasitic. So the answer for P, saprophytic, and R is parasitic. So epiphyte are autotrophic. So the answer is A. Question number 6. If a plant is experiencing chlorosis, it might be deficient in which of the following micronutrients? A, calcium, B, copper, C, iron, and D, boron. Okay, we have explanation about this one, and the answer is B. Okay, look at the next slide for explanation. Okay, deficiency symptom. Okay, elements such as calcium. Okay, what is the symptom? Plant growth is stunted. Irregular shaped leaves, tips of stem and roots die. Copper, chlorosis. Okay, so the correct answer for previous slide is copper. Copper, chlorosis, and abnormal growth. Iron, yellowing between the veins on youngest leaf, but veins remain green. Boron, inhibition of cell expansion, death of meristem, and reduced fertility. Number 27. Diagram below shows a condition of a stomata. Which of the following factors causes the stomata to be in the condition is in diagram below. Okay, look at the diagram. The stomata open. What are the factors cause the stomata open? Look one by one. Okay, the first one, high intensity of light. Correct. When light intensity high, the stomata will open. Photosynthesis occur. And number 2, high relative humidity. When relative humidity high, stomata close. So the rate of photosynthesis low. Number 3, high temperature of the surrounding. Correct. The higher the temperature, the higher the rate of photosynthesis. So at that time, stomata open. Number 4, high concentration of oxygen in the atmosphere. Oxygen is not the gas involved in photosynthesis. So the correct answer is 1 and 3. So the answer is B. 1 and 3 only. Phytoremediation is the process of using eggs to clean contamination from water and soil. What is eggs? Okay. A. Fungi. B. Green plants. C. Filters. And D. Microorganisms. When we refer to textbook, the answer is aquatic plant. So the best answer A, B, C, D is green plants. So the answer is B. Okay, number 29. Which of the following is a function of cytokinin plants? Cytokinin A. Cytokinin helps in ripening of fruits. Okay, B. Cytokinin helps shoot to bend towards light. C. Cytokinin promotes cell division in areas of rapid cell growth. D. Cytokinin helps to absorb energy from sunlight. Okay, so look at the answer. A. Function of cytokinin helps to ripening of fruit. That is the function of ethylene. So A, wrong. B. Cytokinin helps to shoot to bend towards light. That is the function of auxin. C. Cytokinin promotes cell division in areas of rapid cell growth. That is the correct answer for cytokinin function. D. Cytokinin help to absorb energy from sunlight. That is the function of chloroplast. Help to absorb energy from sunlight. So the correct answer is C. A boy has blood group B and his sister has blood group A. Which is the correct combination of genotypes belonging to their parents? Okay, look at one by one. Question 13, D. The answer D, father BO, mother BO. So, their children only B. They have no A in blood group. So, D totally wrong. C, for answer C, father AA, mother BO. So, their children may be AB, AO. So, they have no B blood group. So, wrong. B, AA for father, BB for mother. Okay, so all their children have a blood group AB. Also wrong. So the correct answer is A. Father AB, mother AB. So their children may be A blood group, AB blood group, or B blood group. Okay, so the correct answer is A. Diagram shows germinating of pollen tube. What is structure of R? R is male gamete or means male nucleus. Male nucleus. Okay, so the answer is C. Diagram below shows a part of a flower at one stage during reproduction. Okay, diagram shows X. Which of the following is structure of X? A. Polar nuclei, B. Pollen grain, C. Synergic cell, D. Antipodal cell. Okay, the answer is polar nuclei. So we can see at the next slide the part of reproductive system in plant. Okay, this diagram shows two polar nuclei, 2N. Okay, pollen grain at the top and 3 antipodal cell, 8 cell, tube nucleus, and 2 synergic cell. Okay, the part of plant reproduction. Question 33. The chart shows the pathway of nitrogen cycle in leguminous plant. Okay, look at the diagram. Okay, we go to L first. Organic nitrogen, leguminous plant, to nitrogen in atmosphere. So the process is denitrification. For J, inorganic nitrogen in soil to nitrogen in atmosphere. So the process is nitrification. For K, organic nitrogen in leguminous plant to inorganic nitrogen in soil. So it is nitrogen fixation. L, denitrification. K, nitrogen fixation. And J, nitrification. So the answer is A. Question 34. Diagram shows 4 organisms belonging to different kingdoms. Okay, P, Q, R, and S. Which of the following is a correct match? Okay, kingdom fungi. S. Kingdom protista. R. Kingdom animalia. Q. Kingdom plantae. P. So the correct answer is D. Okay, question 35. The table below shows the result of a field study to estimate the population of garden snails in a vegetable farm. Example, first and second, number of garden snails captured. Okay, first, 280. Second, 70 marked and 80 unmarked. What is the estimate population size of the snail? Okay, you must remember the formula. Population size, number of individuals in the first sample times number of individuals in the second sample divided by number of marked individual captured. So, the number of individual in the first sample, 280. Then, times number of individuals in the second sample. Second sample, you must total 70 plus 80. So we can get 150. Divide by number of garden snail in second mark, which is 70. So 280 times 150 divided by 70. We can get 600. So we can estimate the population size of the snail is 600, which is the answer D. Diagram below shows a pyramid of numbers P, Q, R, S. Which of the following is true about the organism in the pyramid in the diagram? Okay, look at S first. S. Okay, answer for A, autotrophic. Correct. B, heterotrophic. Wrong. C, tertiary consumer. Wrong. And D, fourth trophic level. Also wrong. Then we look at R, heterotrophic. Correct. Q, heterotrophic. Correct. P, heterotrophic. So, from this diagram, the answer is A. Question 37. Diagram below shows cut of apical characteres which place on an agar block. Okay, the diagram shows charactile, light sources, and agar block. What is the possible result at the end of the experiment? Okay, look at the light source, and from this diagram we know auxin diffuse from the tip through the agar block, which cause the shoot to be bent to the light sources. So A, B, C, D. D is wrong. B also wrong. C also wrong. So the answer is A. Question 38. Diagram below shows two types of variation, X and Y. Okay, the diagram show number of students and characteristic of variation X. Which of the following are example of variation X and Y? X, the characteristic of variation X is continuous variation. Y, discontinuous variation. So we must choose the example of variation X and variation Y. Okay, look at answer A. Eye color, variation X. Continuous variation, wrong. Variation Y, blood group, correct. But variation X, wrong. So we can not accept A. B, X. Continuous variation, weight, correct. Variation Y, type of fingerprint. Discontinuous variation, correct. Okay, C, high variation X. Continuous variation, correct. But variation Y, weight. Weight is continuous variation, not discontinuous variation. So C, wrong answer. D, type of hair. Type of hair is discontinuous variation. Y, high High is continuous variation. So D also wrong. So the answer is B. Weight for variation X and type of fingerprint is variation Y. Diagram below shows a profile of a mangrove swamp. Okay, they have a muddy shore without plant. Zone 1, Zone 2 and Zone 3. Okay, what are the dominant species found in each zone? Okay, the pioneer is Avicennia species and Sonoratia species. So Zone 1, Avicennia and Sonoratia. Zone 2, Rhizopora species. And Zone 3, Bulgara species. So the answer is C. Diagram shows a karyotype of a person with a genetic abnormality. Okay, the karyotype, look at the karyotype diagram. 22 plus X, Y. So the karyotype belongs to man. What is the phenotype of this person? Okay, look at chromosome 23. In Klinefelter syndrome, the male has 47 chromosomes instead of the normal 46 chromosomes in his body cell. This chromosome consists of 44 autosomes, 2 X and 1 Y chromosome. So, the diagram shows Klinefelter syndrome. The karyotype shows the Klinefelter syndrome.